Hello friends, so welcome to day 2 of week 1. So in this lecture we will cover the basics of how the DNA is organized in a cell. We'll discuss both eukaryotic and prokaryotic cell, the genome structure and the high throughput technologies that are now used to generate a lot of big data of biology. Okay, so basically in this class we will brush up the basics of genomics that you have read and try to connect that with the how the textbook data can be used for computational analysis. So we have often heard the word OMS, OMICS, like genomes, metagenomes, and okay. So basically, what do they mean? They refer to something in totality. Okay, for example, if I say genome, so that will mean that all the DNA that is present in a cell, eukaryotic or prokaryotic. Similarly, transcriptome, metagenome. So basically, metabolome, epigenome, metagenome, pharmacogenomics. Okay, so many terms are now there, which are because we are now in the high throughput stage. So we want to explore systems in totality, right? So that is why these kind of omics and OMS are becoming very popular and generating a lot of data every day. So we will see what genome is, what you mean by gene, although these gene concept will come in detail later also, the ORFs and the DNA sequences that are coming out of the genomes. So the term genome was coined in as early as 1920, which at that time means genes plus chromosome, which is a very basic definition of genome, whereas now it includes a lot of things that come up via the discoveries that have happened after 1920 till date. So basically genome is referring to the complete set of genetic material which is present in an organism, which is also inherited, and it will include all the chromosomes that are present in a in the nucleus or in the nuclear region in a prokaryote. Okay, the DNA. Okay, sometimes the genetic material DNA, sometimes it is RNA in case of viruses, for example. So that is also the genome, and it includes so what it includes, like the chromosomal DNA, okay, that will contain genes, the non-coding regions, the intergenic regions, repetitive sequences. So okay, and now basically there is a term called haploid, diploid, and tetraploid in case of plants, or polyploidy. Okay, so basically it means that how many copies of the chromosomes are present, right? So basically human genome is, for example, diploid. It has 2 copies of the chromosomes. Okay, so it all started you can say the beginning of genomics when you discovered the DNA structure. Okay, although the term came as early as 1920, but after the discovery of DNA and even after that, when we started sequencing the DNA, which Sanger pioneered, we generated sequences, and then the science of genomics evolved. So okay, so we will see that how the sequences which are now coming out of DNA are analyzed. Okay, so basically DNA was discovered, and then Sanger gave the sequencing method. So initially this method was manual. So first of all, because this technique was also new, they started with very basic genome, like basic in today's terms, but like a mammoth or gigantic in that time, because they just started the sequencing technology. So basically the first genome that was sequenced was a viral genome, which was only about 3,500 base pair long, which was actually done manually, followed by another viral genome which was about 5.3 kb. It was a bacteriophage 5x174. After that, they started attempting the genomes from all domains of life, and they followed it up with the smallest bacterial genomes, like it was Haemophilus influenzae was one of the genome that was done at the Craig Venter Institute in 1995. At the same time, they were also doing the yeast Drosophila, and also making plans to do the human genome. Okay, so the first eukaryotic genome came in 1995, and that was of yeast Saccharomyces cerevisiae, that contained 16 chromosomes, and it was a big effort at that point of time, and it was followed up by the Drosophila genome. Okay. And at the same time they were also doing the other like genomes such as archaeal, like Methanococcus by TIGR. Okay. So at that time genomes sequencing was so challenging and important and difficult that the papers that were published came in very important journals such as Science, which reported the first bacteria, similarly the yeast genome or C. elegans genomes or the Drosophila genome, and more importantly the human genome that came in the year 2000. And like I will mention in the next few slides that it took almost 22 years to complete it, the human genome. Right. So we have seen genomes, and at this point I should also tell you that there is a term called C value paradox or C value enigma. Okay, which means that the genome size does not necessarily correlate with the organismal complexity. So it could be that the genome is very large or too or very small, but now it doesn't matter that if it is too big then it has to be very complex also. Okay. Right. And what is C value? In the C value paradox, it is the total amount of genetic material which is present in the haploid nucleus, or basically one copy of the chromosome that are present. Okay. So C value is basically the simply the genome size or one copy of the DNA. Okay. So like in this you can see that human genome has 23 chromosomes, it is 3.2 billion base pair, and almost 30,000 genes are there. Okay. Whereas if you see aloe vera, it has only 14 chromosomes, but it is almost, if you see, 5 times more than the human genome, right, and contains like only 2.5 times more gene than the human genome, right. Similarly mouse, Drosophila, there are like you can see that like peacock, it has 69 chromosomes, right, and but the genome size is only 1.2 billion base pair. And when you see like banyan tree, which looks so big, old, and even some trees survive more than 1000 years. Okay, so that tree is only 0.4 billion base pair, right. So 1/8 of the size of the human genome, and contains almost equal number of genes like the human genome. So this tells that genome size does not correlate with the organism complexity. There are repeats, there are non-coding DNA, intergenic regions that may contribute a majority of the DNA, and it may not necessarily have that many number of genes also. Okay, so in the past class we also saw like what type of genomes are there. So basically in there are like eukaryotes and prokaryotes that contain the DNA. In prokaryotes it is the circular DNA, whereas in eukaryotes it is like chromosome, okay, which is present is a chromatin, right. And the type of genomes, if you see, like one is the nuclear genome, the DNA which is present inside the nucleus, and there are extranuclear DNA which is present in mitochondria, and in case of animal eukaryotic cell, and also the chloroplast in case of plant cell, and both of the DNA of either chloroplast or mitochondria have prokaryotic origins. Okay. And similarly in case of prokaryotes, they also contain plasmids, which are like extra chromosomal DNA for them. Okay, so the case of human genome, because it is more important for health and it is our own genome, so we will discuss it in little more detail. So and also like the tools and the analysis methods are also more dedicated for the analysis of human genome and larger eukaryotic genomes. So basically you can see in this picture that they have 23 pairs of chromosomes, and you can see that each pair, like chromosome 1 here, it has 2 copies. Okay. Similarly chromosome 3 has 2 copies, right, and you can see that all the chromosome 23 have 2 copies except the chromosome number Y, right. So okay, so this is like the story of the human genome, which has 23 pairs of chromosomes, almost 3 billion base pairs, containing like 25,000, or we are still discovering more and more genes, 25 and more genes, right? And the first genome that was done by the using the Sanger reads, which was about 800 to 1000 base pairs long. And basically you can imagine that you are trying to cover a very big chromosome into pieces of just 800 base pair long read, right? So basically what you will get is information in bits and pieces. So when you are basically, if I give you only this piece, okay, you don't know where to put this piece. So it was like looking at an elephant, but you know, touching at various parts. So like an ant is exploring an elephant, and it doesn't know whether it is on the head or the tail, or like the ear or the leg, okay. So that is why like larger genomes were very challenging at that point of time, and the human genome came in the first draft in the year 2001. Okay, so now to cover this long genome or large genomes, the earlier strategy was to build a microsatellite map. So microsatellites are repetitive units which are polymorphic also and present in individuals, right? So their position are generally conserved, but their length are not. That is why they are called polymorphic. So they will be present in all, but their length will vary. So what they did was they prepared a microsatellite map of the entire chromosome. Okay, like this is chromosome number 10. So it was divided into various bands, and each band they had these microsatellites. Okay, so the first microsatellite map, the entire genome, came in the year 1996. It was done by Deb et al., published in Nature, containing about 5264 microsatellites, right? And which were polymorphic. And using that microsatellites, now you know that okay, I am standing in this region of the DNA, okay, and these are the various points or milestones that I have to cover. So you can target a region between this or this or this, okay. So this way it became easier to cover the region and also to know that what region we are sequencing, and that is how like the entire genome was divided into several parts, and various countries were and researchers were given the different parts to sequence in their labs, and it was a worldwide effort. All the major labs participated, included billions of dollars, like thousands of scientists, both private and public companies were involved, and based on this they could prepare the complete human genome. And similarly, like these microsatellites are basically the dinucleotide or simple repeats, and similarly there are complex repeats, and their map is also available, map of jumping genes, and these maps are very informative when we do comparative genomics or look at the polymorphism or some individual variations in genomes. Okay, so the first genome draft came in year 2000, but actually it took almost 22, 23 years to complete the human genome, okay, because it had gaps. There are some regions in DNA which in the chromosome which cannot be covered by random shotgun sequencing or right, even primer mocking, and so these regions are difficult to sequence, and it requires very long reads, like which are now produced by PacBio or the nanopore ONT method sequencers, right? So these are required to cover the complete genome and fill up the gaps, but still you know, it is it always has to be a hybrid approach, and by putting a lot of effort after 23 years, basically now we have a complete version of the human genome from telomere to telomere, okay. So like from one end of the other end of the chromosome, you now have the complete genome of the human available for analysis, and I think similarly it will be extended to other genomes, and very soon like telomere to telomere sequencing will become a standard practice. So why it is difficult to sequence a genome completely? Because genomes are very large, they contain repeats, and a lot of regions which cannot be sequenced. Even whatever method you use, there will be some region which got missed. Even if you try a lot of coverage to cover the genome, okay, read it many times also, there are some regions will always get missed and require extra efforts to cover that those regions. Okay, so that is why initially what you will get will be the draft genomes, whereas later you will get what you can complete it, right. Okay, so this is the paper that came for the T2T genome, right, telomere to telomere genome of the human, all the 23 pairs of chromosome. Okay, so basically the human genome, if you see, like 90 out of the 100%, only 25 to 27% corresponds to genes or basically the intragenic or intronic regions, and the other part is the intergenic regions. And among the genes, basically only 2% is coding. That means the information that contains codons and will code for some functional proteins, whereas the rest of that is introns, pseudogenes, or basically the other structure in front or back of the gene, the UTRs, right. And the other part, which is the 75%, generally contains repeats and some regions which are unique to that genome. Okay, and repeats now can be tandem repeat like dinucleotide trinucleotide repeats, or interspersed repeats which are longer in size and distributed throughout the genome, and they do not follow a kind of repetitive pattern, but they get inserted at various places, and they have their own kind of functions and also lead to some kind of disruption sometimes. Okay, so right. So okay, so in this picture what I am showing is like all the 24 chromosomes of human, 22 autosomes and X and Y chromosome, and you can see that with the pink color indicates the number of genes and the green color indicates the base pair or the length of the chromosome, and you can see generally like the number of genes are higher than the chromosome, the number of base pairs on the chromosome, or you can say that they follow a pattern. Okay, that okay, whereas in few cases you can see that this is a different picture, like in case of 18 or Y chromosome. So where you can say that these chromosomes are not very rich in the number of genes. Okay, although their length is more, but they do not contain that many number of genes or same proportion of genes as the other chromosomes. Okay, so when we talk about genes, just to touch, like there are different types of ways, methods to find genes. The genes include both the structural genes, that is the tRNA, ribosomal RNA, or basically RNA coding genes, or the protein coding genes. So both have different methods for to predict such kind of genes, and of course it will be very easier and simpler to find genes in prokaryotes because they do not have introns, whereas in case of eukaryotes it will be very difficult because or like relatively difficult because it has a complex gene structure that includes both introns, exons, and also a lot of regulation. Okay, so like what are like if you see like the DNA sequence that we are talking about, for example, we have a sequence here from 5' to 3'. So you can see that I can start reading this sequence if this is the sequence in front of me from A. Okay, so in that way, if I say I am reading a codon, so basically my first base pair will be A and it will be AGG, and then next will be like GTG, okay, or AGT, okay, and TGA, and like this I will read. If I shift by one base pair, I can start reading by GGT, GTG, okay, GAC, and like that. Okay, so this way I can read a DNA in 3 different reading frames, okay, or consecutive reading frames, right, and in all the 3 reading frames I will get different information, and that is why DNA is so informative. Okay, and similarly if I reverse this strand, for example, this becomes G, C, T, A, T, A, T, like, okay, so I can again read it in this reading frame and then this reading frame and this reading frame, to GCT, TAA, AT, and ATN, like that. Okay, so that means a DNA can be read in 6 different reading frames on both the strands, right? So like one example is here also. So in the first reading frame it will be ATG, which is start codon. Okay, we know ATG codes for methionine is the start codon, and the TA, which is the stop codon. So, and basically in between you can see there are different codons which code for various amino acids like CAA, TGG, GGA, AAT, and like that. But when I change the reading frame to okay, so TGC, like that, so you see that there are some codons, but you know the first start codon came a bit later, okay, followed by a stop codon later stage, and the length of this coding region also was smaller than the previous one, the first reading frame. And in the third reading frame also you can see that just by changing or starting from a different base pair leads to a completely different information in terms of codons, and that will result into completely different protein sequence, right, and different start and end of the gene. So all this which we have seen basically is the textbook information, but we can use this information to develop programs and scripts, okay, and to analyze the DNA. Even like if we know the basics of what is a gene, what is the gene structure, okay, what are repeats, and the other informative or important regions in a DNA, we can write programs and scripts to find those regions. Okay, so now coming to what is a prokaryotic and eukaryotic genome. So you see that this is a prokaryotic genome. Okay, so generally prokaryotic genomes are represented as a circle in a circular manner, because they are circular in shape, and it is easier to represent information on a circle. Okay, because like for example you can see here that there are several concentric circles, okay, and each circle will give me different information. I can plot the information that I want on each of the concentric circles. And also you see that like if this is the like positions from 1 to 100,000, 200,000, 300,000, 400,000, like that, you can see that the genes which are mapped here, okay, they all have it is very limited free space. So that means it is mainly very gene rich, and almost 85% of the DNA in prokaryotes is genes, right, and rest is either the intergenic regions or regions like promoters and other which are required for regulating those gene expression, right. So basically the prokaryotes like to keep their genome compact and include as much information as possible, and which is also important because because for example E. coli divides in 20 minutes. So in 20 minutes they have to make another copy of DNA, also make all the other structures of the cell. So they would like to keep the information as minimal as possible, and only important information will be kept in the chromosome, in the gene, in forms of genes. Okay, and other thing is that like their genes are organized as operons. So there could be they are like many genes may be under the control of a single promoter, and they will be transcribed together, and from one transcript you can get multiple proteins. Okay, they will not have introns. So basically it will that is why they have a very small genome size. For example, the E. coli is 4 million base pair, whereas the human genome is almost 3.2 billion base pairs, right. So okay, so now in this if we see, so as I said, it is circular representation, and so like for example the first circle can represent like the base pairs from starting from 1 to end, okay, and the other circles, say this could be the number of genes, this could be the other pseudogenes, and okay, the GC content. So this way we can represent information in circles, okay, which is easier to see, because now I can say that at this position what is the gene here, what is the GC content here, and like here also you can see that in this region, so GC content is this, and the number of genes are showing here on the forward strand and on the reverse strand, and then this part is showing the number of pseudogenes here, and this is showing the other tRNA and other information in that genome. Okay, so that is why it is easier and better to map. So now the same thing that we were explaining in the previous slide. So it generally starts with position number 1, but how do you know that when you have like a circle or a bangle, basically, that what is the start of that wheel? Like you have seen the wheel in a bullock cart, okay, or in your bike rim, that there are spokes. So how do you know that which one is the first spoke in the wheel? Okay, so like from where the wheel starts. So basically for that there is a gene called ORIC, or there is, which is located at the origin of replication, which codes for chromosomal initiator protein, or DNA gene. So that is labeled as the first gene, and then all the information is arranged according to that gene in the circle. So basically this will be given number 1, and from that the numbering of the rest of the chromosome will be done. Okay, and again, as I mentioned, that the different concentric circle will provide different information. For example, the first one is telling the base pair, second one is telling the GC skew, right, and the third one is telling the GC content, okay, and this one is telling the genes which are present. So this way it will be you can put a lot of information, and this red thing is the pseudogenes, okay, and the tRNA genes and ribosomal RNA gene, like which are mentioned here, okay, right. So so this is the typical bacterial genome map which you will observe in various books, and this you can also prepare. So basically to prepare such a map you will need to have information in the form of a table, okay, where in various rows and columns you will arrange information as per the program or as per your requirement which you want to show. Okay, I will also show a gene table later, right, and then the softwares are there which can convert this information into something like a circular map. Okay, so as I mentioned, the first gene is the starts at the origin of replication, or ORIC, right, and the protein which is present at that position is DNA, which initiates the DNA replication in prokaryotes, because you see in prokaryotes the DNA replication is bidirectional. Okay, so basically it starts from here and ends somewhere like exactly opposite to it, which is the replication terminus, right. Okay, origin and terminus. So basically it moves this way, bidirectional, so that they can do this faster. Okay, and this is a typical gene table as I mentioned. So like for a genome like this which you are seeing here, so it has genes starting from position number 1 and then going this way. So basically you have to write it this way. So gene 1, gene 2, gene 3, gene 4, gene 5, gene 6, gene 7, 8, 9, okay, and they will have a gene ID also, which could be a standard gene ID, or it can be intermediate gene ID which you have generated from your program or for your genome. Okay, the start of the gene and the end of the gene. So basically starts with 1, position number 1, and ends at 1317. So it is 1317 base pair long, and divide by 3 it will be certain amino acids. So it is present on the positive strand, and that information is also important, that on which strand it is. The positive strand, DNA is double stranded, so it is this way or on the other side, right, and the function of the gene which you get by methods of various function annotations which are available. Okay, now another thing that I want to introduce is like when you have genes and they are doing some function in a cell, right. So okay, so basically for example you want to convert, say you want to use the glucose that you are consuming or eating. Okay, so glucose is a sugar molecule, and it will be degraded, okay, into energy, right. So basically it will be done in a molecular pathway, right. So today like there are groups like KEGG, it's a very important and standard database of all the metabolic maps which are available or pathways which are available. So what they have done, they have constructed a comprehensive map of all the pathways. Like you can see that like glycolysis starts here, okay, and then it goes till pyruvate and here, and then acetyl coenzyme and moves into the TCA cycle and like that, okay, and again the electron transport chain. So basically the entire information is mapped in pathways, because pathways are made up of intermediate steps where there are intermediate reactions which are carried out by these specialized enzymes, right. So this way we know that what all pathways are present in a genome, and we can map the genes which we find in a genome on the pathways to see that okay, this pathway is present on this pathway is not. It can, okay, for example in this picture you can see that the previous one is all colored, and you can see that all basically this is a node and this is an edge, the line that connects a dot or a circle, right. So nodes and edges are all colored, but here if you see it, there are many pieces which are blank, okay. So basically these pathways are not present in this genome. So what you can do is you can take a reference map and map your genes on that reference genome, and then you will know that what all pathways are present in that genome, okay. For example, I can say from here that it contains glycolysis and also TCA, okay. So just by looking at this, and we can tell that okay, this does not contain these pathways but contains these pathways, okay. And when you do the KAG analysis, it also gives you information about all the pathways separately also. This is a comprehensive picture, complete one picture, but you will also get pathway information for this part and like that, separate pathways for your analysis. And then when you publish a genome, you have done the gene prediction, you have done the pathway mapping and analysis, and after that you know that okay, the genome that you have in hand is basically can produce which amino acids, what is going inside the genome, okay, what is going outside the genome, like this is producing ethanol and acetate and D-lactate which is going out of the cell, and what it is taking out from outside the cell, right. So from the outside environment. So this way you will know that what the genome is doing in that environment, or if it is like endosymbiotic or symbiotic, what it is producing for the host, or what it is taking in from the environment and what it is producing out to the environment, right. So now when we talk about genes, we should also understand that genes are not present in a single copies always, and there could be multiple copies of the same genes which are called gene families, which arise because of duplications. For example, like you have one gene, so now duplication events are do happen in genomes over a period of time, right, and in recombinations and other by other repetitive elements also. So now, so basically by the duplication you can see that this gene which was like A gene becomes A1 and A2. So now two copies of the gene A became emerged, which is A1 and A2. So okay, so now A was doing some function, okay. So now when you have two copies, for example, now A1 continue doing the same function, but if I change A2, it will not impact the organism because the function which was supposed to be done by A1 is still happening, okay. So now A2 can diverge or change, okay, over a period of time, and that will not impact the functioning of the original A or A1 gene, okay, but it can lead to new function origin, right. So it can lead to new functionalization by basically this gene changed, okay, this copy changed, and right, or basically sub functionalization that the two copies have their own specific role. It could be tissue specific, or it could be the developmental specific, or even if one of these copies is lost, for example in this case, it will not matter, and still the genome can continue survival by using only one copy. So gene duplications are very important events in the evolution of an organism, and whenever we genome sequence a new organism, we always do the analysis, comparative analysis, to see the gene duplications and the expansion and contraction of gene families, and which is very good indicator of what and how the genome evolved over a period of time. Okay, the other term that I want to introduce is a pseudogene. Okay, so we have seen that a gene is there, but it is possible that a gene can get disrupted or lose its function. How? Because like the promoter sequence gets some mutation, or the start codon gets some mutation, or there is something like we talked about retrotransposons, or right, so they may come and get inserted in between, or basically during some of the event a part of the gene is lost because of the recombination event, or or breakage of the chromosome. So a gene can lose its function by these manner, and okay, so this is a gene which actually is making the RNA and the protein, right? But what can happen is, like in case of, say, which we discussed in the previous slide also, a duplicated genes, two copies of the same gene, okay, one copy remain function, another copy get a mutation. So you have now a functional gene and a pseudo gene which has lost its function because of some random mutation that has happened. Or it could be sometimes that the RNA which is made goes back and insert to the DNA and forms the process pseudogene. So pseudogenes are also present in genomes, and they may look like the real gene. Okay, like for example, this may contain all the other elements of a real gene but has a premature stop codon. So they will also complicate your gene prediction analysis and need some manual effort to find such pseudogenes in your analysis. Okay, so now after genes, the rest of the part of the genome is mostly repetitive, as we see, like more than 55% of the human genome is repetitive. Okay, and which composed of these simple repeats, like for example TG, 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 TG. These are simple dinucleotide repeats, or CAG, 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 okay, polyglutamine repeat, like that. Okay, or it could be, so basically these are small tandem repeats, repeating units, okay, CAG, CAG repeating one after another over a period of, for a long length. Okay, or there could be complex repeats like signs, lines, okay, or major segmental duplication, the long regions of chromosome which are duplicated, the transposons. Okay, so all this comprise of the repetitive sequences in the human genome, and only 2% of the genome is actually coding for the functional proteins, and the remaining after repeats and the unique regions is the intronic region, which is basically present in the genes but not actually the coding regions. Okay, so now just an overview of what are repeats. So basically there could be interspersed repeats, okay, like these, or there could be tandem repeats. Okay, so we will see them in detail. So these are tandem repeats, so they are repeating units, like I can mention TG, 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 like that. Okay, 6 or more, okay, in a genome. These are dinucleotide tandem repeats, or dinucleotide repeating units, simple repeats. And the other one could be the complex repeats, so which are not tandem. Okay, so one unit can be here, another unit is here, or maybe this unit came because this made a copy of itself and got inserted here, right? So simple and complex repeats are present in the genome. Okay, like this is the example of a complex repeat, like retrotransposons, which make a copy of themselves to RNA and then again go back to DNA and get insert. Okay, and this way wherever they get insert, so they may disrupt the reading frame, because we saw that DNA is very particular about the reading frame. So I can start reading the DNA from position 1, 2 or 3, and in 1, 2, 3 reading frames I will get a completely different codon structure, right? And ultimately the gene or the amino acids sequence, right? So if they get insert in between that reading frame, they will disrupt. They can also disrupt the splicing pattern by getting inserted into the canonical splice sites, or they can also, the chances that they can get insert in the some of the repetitive regions itself, and which should not impact the function, right? So signs and lines are very common repetitive complex repetitive sequences in the human genome, and like particularly the allele repeats, which are very important part of the human genome and constitute almost 30% of the human genome, and also do a lot of functions, and also important for the evolutionary context. Okay. So we have also already talked about lines, which are repeats which make copy of themselves and get inserted in various places and may disrupt the function, right. And for example, in case of colorectal cancer, they get inserted into important gene and disrupt its function and may lead to cancer. Okay. Also lead to evolution of new genes because of their random insertions. They may help the genes to gain functions, and we are still trying to understand the role and function of these repetitive elements in the human and other genomes, particularly in plants, which is almost like 70% of the plant genome is repetitive. Okay. And there are other like short repeats like tandem repeats, which are also called microsatellites many a times. So like you can see that this is a part of the genome. Okay. So which is written in the like 60 characters per line, and each block you can say is 10 base pair long, right. Okay. And you can see that in this region there is GT, 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 and there is a long repetitive unit here. So this is an example of a tandem repeat. So this is dinucleotide, and similarly there could be trinucleotide or CTG, CTG. Okay. And particularly you can call it as CNG N, right. So N refers to number of times CNG or this CTG or CAG repeat is repetitive. Similarly, if I want to write this, it will be written as GT in bracket N. N will tell how many copies are there, like 1, 2, 3, 4, 5, 6, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So in this case it will be GT20. Okay. So this is an example of a tandem repeat, and they are very important for gene expression and have been associated with variability in gene expression because of their location in the gene, that is generally like upstream of gene, and sometimes also in the in between the genes, in between the gene, right. And there is one more important repeat which is called telomeric repeat. Okay. Which is like shown in this case, like this is one telomeric repeat, right. Okay. You can see that this is a long repetitive unit, and this is a human telomeric repeat, and these are present at the telomeres, and these are they can adopt a very complex G quadruplex structure, and which is a alternate form of DNA, and actually this is very important for the conservation of the chromosome at the telomeres. And when we have seen so many repeats, there has to be some program to find the repeats, and one such very important program and commonly used is repeat masker. So this program is very old but still evolving, and this can help to detect both the interspersed and simple repeats in the genome, and it can also mask your DNA and for the low complexity region. So basically these repeats which I have mentioned are called low complexity regions because they are non-genic regions generally, right. So they do not code for the proteins, so they are low complexity regions, and this repeat masker program can help you to mask these regions. And okay, for example, this is the complete gene chromosome, and this was a repeat. Okay. So this will put N here, so this will mask this region, right, and you will get the remaining for your analysis. So this way your gene finding programs or any program that you like to find for various features in the chromosome, functional features, will not get confused, or even computational effort will not be wasted if you have already masked the repeats, and which we do many times when we do the genomic analysis. And this is available at repeatmasker.org. Okay. And finally, like it is important to understand that there is a variability in DNA in all of us. Like if I say that even in brother and sister, you can see the DNA is 98.98% identical, and in the coding region, that is 2% of the genome, it is 99.999% identical. So not exactly identical. So for any unrelated individual, like you all are attending this course and you are maybe unrelated to each other, that means you do not belong to the same family, okay, or do not have any overlap in the family history, okay. So basically you can see that in that case you will be 99.8% identical with each other, and in the coding region you will show a little higher identity, okay. Now this is between the human, which are like human to human, it will be like about on an average 99.8% identity. But if I compare human and chimpanzee also, I can see that 98% of the region DNA is identical, right, which is a very large level of identity, because considering that the chimpanzee and human look so different. So basically you can say that just the 2%, okay, of the genome explains that why the two things or the two genomes are so different from each other. And similarly, like if I say baboon, which are they have almost 300 million base pairs different, they will be only 92% identical to human genome. So basically what it tells, like I can go far also, like mice only 90% identical, spinach which is that you eat like palak, so basically it is only 40% identical. So still like a human and a spinach shows almost like 40% identity. With mouse you show 90% identity. So which shows that there was a common origin for the DNA, and later on it got evolved and got differentiated into various functions. And the study of these genomes or related genomes, unrelated genomes, will tell you how a particular character or phenotype evolved in a genome. And we get several clues about the health, the origin of disease, or a special feature or character in a genome by doing this such kind of comparative analysis. And also like these genomes like chimpanzee and baboon, okay, and are very important for medical reasons also, because for example you want to see that why, like you want to try some medicine or drug, or okay, then you can use these as model organisms, right. And also you can see that okay, there could be some disease which is happening in human but not happening in chimpanzee. So you will know that by looking at the difference, okay, you can see that why what evolution or what change happened at the DNA level, molecular level, and also at the cellular level, that one disease is infecting the human but not the chimpanzee, and vice versa. So these things are very important medically, and we want to generate information. For example, recently the chimpanzee chromosome map is also prepared, and you can see that both have equal number of chromosomes, but only difference is that like the chromosome number 2 of human, so basically which is divided into two parts or split into two parts in case of chimpanzee, 2A and 2B. Okay, earlier it was counted as two different chromosomes, but later on it was realized that okay, no, these are the two parts of the chromosome 2, and then it was rearranged into the same numbering of chromosome 1 to 22, and then X and Y. Okay, so we see that genomics is one important area. We generate information from DNA. Okay, so then how do you generate this information? What are the new technologies? Okay, there are so many actually, but I will just touch a few of them. Okay, and they will also come later in detail, okay, which are producing this big data of biology, okay, and which are very important for the genomics. For example, for sequencing, there are multiple types of sequencers not available, or you can say the sequencing technologies. For example, the Illumina technology, which produce short reads, and short reads but deep sequencing. That means you can get like several hundred times coverage for an individual in a given run of Illumina. Similarly, there will be long read sequencers where up to like 25 to 100x coverage for an individual will be more than sufficient to cover the genome. Although these may suffer from some errors, okay, for which you can generate reads from here and map to this, and this way you can improve the quality of the long reads, right. So that is why hybrid sequencing is important these days, right. So there are different platforms like Illumina short reads, PacBio, ONT long reads, okay, and 10x Genomics, which are important to basically link reads for mapping or basically preparing a better and contiguous map of the genome, okay. And now like PacBio hi-fi reads have come, which are long as well as highly accurate, which are but the technology is still expensive, but these are very important to completely cover the genome, and basically for the telomere to telomere map of the human genome also PacBio or ONT played a very important role, the long reads, right. And there is also a Hi-C technology which captures the spatial conformation of the chromosome, and this helps in constructing the complete chromosome. Otherwise, when you extract DNA from a cell, it is basically you don't know what chromosome you are actually handling, okay. It is only DNA, a mix of all the chromosomes, and later on by Hi-C and other methods you can know that you can divide that into various chromosomes, the information, okay. So hybrid approach or hybrid sequencing is the way today to do the sequencing of genomes and to make a contiguous and complete genome, okay. And the other one is the nanopore reads, which may be error prone but much cheaper and easier to do. For example, this is a sequencer like a USB, so you can insert in your system, okay, one cable from here, right, and this will be USB end. You can insert this in your system and you can do the sequencing right away. And okay, similarly a small like 7 by 7 inches small device in which you can load your DNA and do your sequencing, and no computer also is needed in this case. So these will be much cheaper and faster way of doing, okay. So there are different versions and varieties of sequencing platforms available these days, okay, which can generate data for genomes, transcriptomes, epigenomes to find the polymorphic repeats, polymorphic regions, SNPs, ESTs, molecular markers, exomes, metagenomics, resequencing of genomes. Like although we have done the human genome sequencing, but when you want to know about a particular individual, as I mentioned that it will not be exactly identical to the reference sequence, and every two individual will be different in the DNA. So you have to do resequencing at that point to look at the genetic variations and for the phylogenetic studies. So all this you are generating a lot of data every day. Similarly, microarrays. So one way to capture the expression, that how much a gene is expressing in a cell, okay, one way is to read all the transcripts which are present in a cell by transcriptomics, right. And the other way is using a microarray, because if I know that what all genes are present in the human genome, I can devise a micro array catalog, and basically you for the selected genes or for the complete set of genes which are present and in a individual or in a organism, and look at the expression level of those genes. So microarrays also produce a lot of data, and the transcriptomics, including transcriptomics, which is there in the databases, and okay. And for example, like for any genome you generate genomic as well as the transcriptomic data for the analysis. And the other prominent player here is the mass spectrophotometry, which basically is very important and powerful method to know that what metabolites are present in a cell, because one is gene, which has the information, then transcript and the protein, but actually proteins are like many times enzymes, and those enzymes produce metabolites by various reactions. So you want to know in real situation that what is consumed and what is produced, so that you can know by mass spectrophotometry, okay, which could be GC, MS, gas chromatography, or liquid chromatography, or it could be the NMR based approach, which also are producing a lot of data for the biological analysis. So basically these days people, for any project, they would like to generate the genomic, transcriptomic, metabolomic, metagenomic if microbes are also involved as a component of or for that system. So data from multi-omics and then do the analysis. Okay, so as we see here, basically the data is doubling every 7 months, and to cope up with such rising and increasing amount of data, you have to develop new and new methods of analysis, faster algorithms, a lot of high computing systems, high performance computing systems. And as you see, like, why this is happening, because the cost is also going down. So if the costs go down, the data will go up, right, because it becomes more affordable and everybody and anybody can do sequencing, right. So like in COVID recently, we generated so much data about the variants of COVID virus in in various regions, individuals and across the world, right. So, and which also involved a huge amount of computational exercise to understand the origin, changes and drug targets in the genome, in the viral genome, right. Okay, so basically this demands for a lot of computational analysis to do computational genomics. And in the next class we will also discuss about, so we have seen data, we have seen the basics so far, like the genomics basics, right, and we know that data is being generated for various projects and objectives and aims. So now how that data is stored, organized and can be used, right, this we will see in the next class. Okay, so in this class what we learned today is how the genetic information is organized in prokaryotes and eukaryotes, what are genes, genomes and chromosomes. Okay, how the genomic maps are prepared for human or for bacteria or the prokaryotes, right, the gene structure and the technologies that are generating the mammoth amount of sequencing and omics data from using various methodologies and platforms. And so we will now meet in the next class, where we will look at how this data is organized and how you can retrieve this data for the analysis. Thank you.